I'm going to talk to you about using conformal prediction for novelty detection in microfossil analysis today. So I can just mention that I'm a PhD student at Visual Intelligence located in Tromsø, and that this is a collaboration between UiT and Equinor and Aker BP. This work been going on for 3 years, involves me, Benjamin Rigaud, and Fred Gottliebsen at UiT, David, the moderator, Equinor, and also Odd Kolbjørnsen at Aker BP. See here, okay. Yeah, so the outline for this talk is 3 parts. A little bit about background, about microfossils, data, challenges, and applications. Second part is self-supervised training of a vision transformer. And then the third part, which will be the main part in this talk, is about conformal prediction. So a little bit about microfossils first. So fossil is a remain of past life, and a microfossil is a microscopic fossil with a size of less than 1 millimeter. So you study them through a microscope typically. This can be complete microorganisms. A foraminifera is a type of microfossil that is extensively studied at geology here at UiT, for instance. But this can also be organic remains of larger life forms, such as pollen from plants. An enormous number of different types and species of these fossils. They are found in abundance globally in marine deposits, for instance, when you drill wells. Very important in a field called biostratigraphy, which involves correlating sediment layers, matching layers across different geological sections, such as between wells. So they are used extensively in subsurface analysis related to oil, gas, carbon capture and storage, but also in research to understand past climate and evolution of plants and animals. And the data that we work on, they are produced by the Norwegian Offshore Directorate. So drill cuttings are reported to the NOD, and they treat the sedimentary rock by a chemical procedure. You get a liquid at the end that you disperse on a slide, glass slide, that you again feed through a high resolution scanner to create images of the data. So what we work with looks something like this: high resolution slide image with lots of organic fossils, possibly tens of thousands of fossils in one slide. And we have very large volumes of this. Well, at the time of currently about 50,000 slides from 430 wells, maybe as much as 3 billion fossils in total, who knows? Yeah, hosted online, and of course any kind of manual analysis of the data is extremely time consuming, and lots of the data will never be analyzed. So you need to automate this somehow. And related to this, we propose to make a pipeline for automatic microfossil analysis using object detection, self-supervised pre-training, and then to create a model that you can apply for downstream tasks. So part of this work was published last year. You can say that the remainder of this talk is a continuation of this work. I can just mention a couple of applications that you can use a microfossil model for. One is in context of content-based image retrieval to locate indicator species, for instance. So you have a query, and then you can feed it through the foundation model, compute the distances in the latent space between the query and all the data that you find in a source to retrieve nearest neighbors. Another application which is more relevant to what I'm going to talk about today is in biostratigraphy. So to the right, you have a stratigraphy report where you have it's not so easy to see, but you have different geological time periods to the left corresponding to different depths in the well. And you have on the sort of x-axis, you have counts. So the blue bars are counts, and you have sort of this vertical names. They are many different microfossil species. So so in a stratigraphy report, you have these species count distributions across depth and time. So this is one application that we sort of want to investigate in this work to try to create these distributions in a more automatic fashion. Yeah, so I want to talk a little bit about the self-supervised pre-training also. So say that we need to we have a lot of unlabeled data. We need to create the training data by creating automatic bounding boxes. So we have investigated a couple of approaches, both like deep learning and also conventional digital image processing methods. Say that quickly. We just the slide is very high resolution, so we handle this by slicing it up into tiles of more manageable resolution, and then process each tile sequentially. And then we generate the automatic bounding boxes by a couple of different methods. We have tested out YOLO V5 pre-trained, not for labeling, but just to get the bounding boxes. And we have also investigated more classical image processing by blurring the image, merging some of the channels, thresholding, and then using connected components to generate region proposals, and then filtering the proposed regions by an area. Both of these approaches work really well. When generating the training data, we have actually used this connected component approach, and we have sampled data. So they're sampled training data from 338 different wells, over 9,000 slides, so over 3 million fossils from a range of wells, which is a somewhat statistical representative data set, although it's like a very tiny fraction of all the available data out there. And the generated data set contain crops of very varying quality, lots of image artifacts, objects out of focus, blurry crops, stuff like that. So we also have an additional data curation step, where we use a pre-trained vision transformer. It's pre-trained on Dino, on the ImageNet dataset, to generate embeddings, and then use a k-means clustering to cluster the data, and then manually by visual analysis, taking out clusters of really bad crops, as you can see in the top figure here. So in the end, we have a data set of about 1.6 million crops with really nice quality, as you can see in this random sample from the curated training data in the bottom figure here. Yeah. And then we use this to train vision transformer self-supervised. We use this DINO framework, knowledge distillation with no labels. This is a very popular method for representation learning. Presented in 2021. In this framework, you create different views of same image, and then you have two Siamese networks that learn from each other to create the same representation of all the views. So there's a teacher and a student network with the same architecture, but with different weights. I'm going to, I think in the interest of time, I'm not going to go into detail about this DINO, how this DINO framework works. I can just go through the results and say that we train both a small vision transformer, UiT-S, and a WIT base using these 1.6 million curated fossils. With this framework, and then we evaluate by with an additional labeled data set that we have that contains 20 different classes. We evaluate both in the context of content-based image retrieval and also nearest neighbor classification. So the results that I have highlighted here are from the nearest neighbor classification F1 score, which is harmonic mean of the precision and the recall. So the red squares are the best pre-trained foundation model, which is the DINO model trained on ImageNet, and the green squares are the models that we train on our data. And our results are really good. We get substantial improvements compared to existing models. So get really good results. So that's sort of concludes this part. I'm going to move on to the conformal prediction part. So going into a little more detail here. So I'm going to talk about conformal prediction with an example, just in general, and then some motivation challenges related to the microfossil data and how we can use conformal prediction for novelty detection. And I will present some results at the end. So conformal prediction is an uncertainty framework for prediction models. So typically regression or a classification scenario where you want to create a prediction set or an interval with statistical guarantees. So instead of a point prediction, you want to create a prediction set that has a statistical guarantee of covering the correct value. So this is something that was first published in 1998, Kammerman, Wolf, Kvapnik, developed since then by many more. And I'm taking this example from a really nice tutorial by Angelopoulos and Bates, which is worth a read. So in this example, you assume that you have a trained predictive model F for K classes. So a classification model, which is trained on some training data. you assume that you have some set of additional data from the same distribution, which you call the calibration data. And then you have a test case X star, and you want to create a prediction set for X star such that the probability that your prediction set is containing the correct class is at least 1 minus alpha, where alpha is a chosen error rate or significance level. And you can actually achieve this by the following steps. So first, you need to calibrate on your calibration data. For instance, you can compute the softmax for the true class on the calibration set. And then you define a conformal score, S, which is 1 minus the softmax. So the conformal score is sort of a heuristic notion of uncertainty. The higher the score, the higher the uncertainty. And then you compute this conformal score on all your calibration data. And you define a threshold, the Q hat, to be the 1 minus alpha quantile on the conformal score distribution. So now you have calibrated your Q hat to the calibration data. So when you do the prediction, you compute the softmax for the test case X star, and then you include in your prediction set all the classes with a softmax output that is at least 1 minus Q hat. And this procedure is actually guaranteed to provide what you call marginal coverage, as long as the calibration data and your test data are from the same distribution. So that is, of course, an important assumption. So a marginal coverage means that this will be true in general, but it might not be true for any particular group or class. So you can, but anyway, you can interpret this conformal prediction framework as where you have this conformal score S, which gives a heuristic notion of uncertainty. Then the conformal prediction sort of transforms the heuristic uncertainty into rigorous uncertainty estimate. Yes. And also note that conformal prediction doesn't put any requirements on the classifier or the score function. In principle, they can be arbitrarily bad, which might sound too good to be true, but of course, if that is the case, the only result will be that your prediction sets will be unacceptably large and may even contain, all the classes in the class taxonomy of the classifier. So then it won't be of any help. Yeah, and as mentioned, marginal coverage only guarantees that you will be correct in general, but does not provide any guarantee for any particular class or group or partition of the data. So in reality, when you use conformal prediction with a marginal coverage, you will likely overcover easy groups and undercover hard ones. So usually what you would like to achieve is conditional coverage, which is a stronger requirement, so that if you condition on one class, then your prediction set will contain the correct label with a certain guarantee. So to achieve this, you need to treat each class separately. Yeah, so that was an example of using conformal prediction. Now I'm going to go back to our data. So from before, we used self-supervised learning to pre-train a small vision transformer to use as a foundation model, so to speak. And we also have this additional 20 classes, which we use to train a linear classifier on top of the vision transformer. Now we want to apply this to some test data, and the wellbore data typically involves slides at different depths. So for one well, you might have anything between 50 to 300 slides for different depths. And we would like to apply this classifier to create species distributions across the depth. And we focus on these 4 classes in this analysis: this Inapertura pollinitis, Paleoperididium, Bisacates, and Alisacysta. Yeah. So a challenge when we are applying the classifier to well data is that the wellbore content will include lots of what you call auto-distribution crops. So imaged artifacts, inertinites, objects that are out of focus, but it can also include a genus that is not labeled in the training set. And you would like to generate a prediction set that identifies most of the in-distribution data in the well and that do not contain too much auto-distribution data. And also, very importantly, that the prediction set is statistically representative in terms of relative frequencies. So we might not detect all the crops in the slide, but the relative frequencies should correspond to the true distributions. And yeah, we define in-distribution data in this context as belonging to one of the 20 classes that we have in our label, the training set, and everything else we label as auto-distribution. So to translate this to statistics, we would, if we define an in-distribution crop as a positive and an auto-distribution crop as a negative, we would like to control the false negative rate. That is the probability of labeling an in-distribution crop as auto-distribution. We want that to be no larger than some chosen value alpha. We also want to control the false positive rate, so the probability of detecting auto-distribution crop as in-distribution, want that to be lower than some beta, and that the false negative rate is equal for all the classes. And we approach this by using the entropy as the conformal score. So the entropy, so when you do a prediction, you get softmax for all the classes, and then the entropy gives a single number that serves as a measure of uncertainty of the classification. So in this figure here, you have the entropy on the x-axis, and then then the blue curve shows the distribution of the entropy for auto-distribution data. So we have predicted a lot of auto-distribution data, computed the entropy, and then we have this distribution. And equivalently for the orange curve, when we have labeled in distribution data, we can make predictions, compute entropy, and generate that curve. And then our goal is to find a rejection threshold Q, which is the black line, so that the alpha corresponding to some value alpha. Yeah, and we would like to do that for all the classes. So how do we find this threshold? Well, we need to ensure conditional coverage on the classes. There are 2 ways to make a type 1 error. So you can either the classifier can be correct in the classification, but the entropy could still be so high that you falsely reject a sample as an out distribution, or the classifier could be wrong, and which implies that we need to take the class-wise recall into account also. So finding the threshold, you can find that by a small derivation. So you know the false negative rate should be lower than some alpha, and when you do the math, you end up with a Q that is the 1 minus alpha divided by the recall of class J quantile on the entropy distribution for that particular class. And then you repeat that for all the classes that you're interested in. Note that we also want to avoid type 2 errors. So probability of type 1 error controlled by alpha. We can also compute the probability of a type 2 error, which we call beta here. But note that if you have a pool of test data and a number of auto-distribution images in that pool of data is large compared to the number of in-distribution data, then the false positive rate can be really small, and you can still get a lot of auto-distribution data in your prediction sets. So therefore, we instead use the false discovery rate as a measure of the quality of the prediction sets, which is the estimated fraction of auto-distribution data in your generated prediction sets. Yeah, and false discovery rate can also be calculated exactly by a small derivation. You get this expression 1 divided by 1 plus 1 minus alpha over beta times r. So where r is a ratio of in-distribution data compared to auto-distribution data in your test set. So the false discovery rate is influenced by 3 things. So the separability of the distribution, so how far the in-distribution entropy and out-distribution entropy is, the noisiness of the data, that is the R ratio, and also your chosen significance level, the threshold. So by increasing alpha, you move the threshold to the left, thereby reducing the number of false discoveries. Yeah. In practice, we have to determine the entropy distributions somehow. So for the in-distribution data, we use the label data that we have. We train an ensemble of 10 different classifiers using an 80-20 train calibration split, and then we use the calibration data as an empirical entropy distribution for the label data. regarding the auto distribution data, we really don't have a data set that with images labeled at auto distribution. And so what we instead do is that we take all the crops in one well, make the predictions, compute entropy, and use that as a proxy for auto distribution data. And in that, we just assume that most of the data in the well will be auto distribution. There is some challenges connected to this because not all the data will be auto distribution, and the left tail of the auto distribution entropy will be dominated by in-distribution data, leading to an overestimation of the false discovery rates, which you sort of illustrated in the bottom figure here, where you have alpha on the x-axis, false discoveries on the y-axis, and you expect that when you increase alpha, the false discovery rate should decrease. But in this example, it actually increases at the end. So to overcome this these sort of left tail issues, we fit a normal distribution to the data, where we get curves that are much more realistic. Yeah, and I'm going to present some results here at the end. So we restrict the analysis to these 4 classes here, partly because they are visually very distinct, and also because they achieved almost perfect F1 score in the evaluation. So you know that the classifier is supposed to do a good job for these classes. When we want to estimate the false discovery, we have to actually guess the R ratio, which we can't really know. So one way to do that instead is to use different values and make different curves, as here, where like we have a very optimistic, conservative, and very conservative estimate of the relationship between alpha and the number of false discoveries. So while it's difficult to at least make a prior accurate estimate of the number of false discoveries, we can see, for instance, that like the green curve, which corresponds to in Apertura Polonites, will likely generate much fewer errors compared to the red curve, which belongs to the Paleoperidium class. And then we, yeah, so to assess our method, we create prediction sets for each class for 3 different thresholds. So alpha equal to 0.05, 0.50, 0.95. And we provided data to an expert at Aker BP, Manuel Vieira, who took a random sample of at most 300 from each of the set, and simply assessed whether the prediction set actually contained the class of interest or not. The actual error rates corresponded quite nicely with the conservative estimate of the false discoveries. You can see, for instance, in the Apertura Polonites class had very, very few mistakes at alpha 0.05 and no mistakes after that. Paleoperidium was much worse. 80% were actually auto distribution at low alpha. But then, of course, the prediction set improved when you increased alpha. So sort of a conclusion from this is that by choosing a significance level of 0.50, which means that we basically discard half of the in-distribution data in the test, will lead to prediction sets that are roughly correct and that will contain very few out of distribution crops. Yeah, so we use this to create this count charge species distributions for 2 different wells. These distributions here, we do not have any ground truth for these distributions, but you know from the evaluation in the last slide that at least our prediction should, the set should contain very few errors. So any sort of analysis of these would have to be done by a micropaleontologist, I guess. But you know, you can see, for instance, for the Inapodurable class and the Paleopteridinium class that you have a spike at around 1920 on the left and 2,600 meters in the right plot. So maybe this is an indication that sort of 1920 meters on the left corresponds to the same geological time as the 2,600 meters on the right, but who knows? Yeah, and also have a slide that where we actually do have a ground truth. So I threw this in last minute, so I don't have any pictures of the classes that we are actually looking at here, but we have a stratigraphy report for a ground truth, which is on the left-hand side, and our the curves that the distributions that we produce with this method are on the right. And yeah, at least from the Rigodella species, it seems that we are at least onto something, even if the distributions doesn't really correspond perfectly. So yeah, this was the final slide, I guess. Some conclusions. I think this conformal prediction framework is a really promising approach for creating statistically representative samples in these kinds of situations where you have a lot of auto-distribution data in the mix. You can get a very quick high-level analysis of the wellbore contents. And of course, lots of challenges and future work related to this. How do you create entropy distributions accurately? Distribution shift is a possible challenge. Is the distribution the same when you move from one well to another? Likely not. You can also connect this to other entropy uncertainty methods. You have some methods that try to enforce a high entropy for auto-distribution data, low entropy for in-distribution data. This can be connected to this, probably a lot more things also. But yeah, that's it.